So, hello friends, I am Dr. Gurpreet Singh from Department of Biotechnology, Delhi College, Jalandhar in Punjab. And today I am going to discuss about the topic the enzyme classification and nomenclature. So, this topic is very relevant as well as the theory and the research is concerned, since we all are concerned with the various enzymes, whether in the research or in our daily life. So, how to classify or what is the method of classification as far as latest knowledge is concerned? So, today I am going to discuss the topic with the following content, that what are the various types of chemical reactions that are proceeding in the living system. So, that means before we move to what is enzyme, what is the relevance of enzyme, what is the significance of enzyme, and how we classify what is the different types of classes that are there in the enzymes, we need to understand what are the various types of chemical reactions that are proceeding within the body and why we really need enzyme. What what is the logic? What is the scientific notion behind it? That why the chemical reactions that are there, they are in the living system, they are not proceeded under the normal conditions, and what they need the various enzymes. And then we will discuss about the history of the enzymes, the history of enzyme and enzymology as such, and what are the latest IUPMB nomenclature and their classification. And in the end, I will basically describe the various examples for each class before concluding. So, coming to the very first component, that what are the various types of chemical reactions that are proceeding in the living system, because we all are knowing that there are various metabolic pathways that are going on, and in each of these metabolic pathways there are so many biochemical reactions that are proceeding. So if we look at the table, we will find that there are not one but there are so many types of biochemical reactions that are going on within the reaction. Or you can say these are on a chemistry point of view, they are nothing but either like let's say the oxidation reaction. So it is nothing but a sort of electron transfer reaction that is proceeding. So what is called the oxidation reduction, or they are generally known as the redox reactions, right? Or we can say there are ligation reaction. Ligation is the general means the formation of a covalent bond. So this is again a sort of a chemical reaction that is requiring the ATP cleavage. So here the energy is provided by the ATP, but when we talk of the chemistry, that means those reactions which are involving the formation of the covalent bond, whether it's a carbon-carbon bond or carbon-oxygen bond or whatever. So they are called the ligation reactions. Then there are rearrangement reactions in which the one functional group they rearrange it. So within the molecule get rearranged, let's say from cis configuration to trans configuration or something like that. So so that is called known as the isomerization reaction. Fourth category in this is called the group transfer reaction, in which one functional group gets transferred from a reactant to the product. Right. So it is nothing but the transfer of the functional group from one molecule to another. So this is called a group transfer reaction. Similarly, there are examples of the cleavage of the bond or the by the addition of the water molecule. That is called the hydrolytic type of reaction. And another category is the addition or removal of the functional group, leading to the formation of a double bond or the removal of a double bond. So these are the various types of chemical reactions that are proceeding within the body, within the living system. And if we look at the chemistry aspect, then these are nothing but the movement of electrons or the transfer of the functional group. So the question that comes to the mind is that why these types of reaction they are not able to proceed in a proper way without the involvement of enzymes, or we can say what is the relevance or what is the significance of these enzymes in the living system for carrying out these reactions. So that what we are going to discuss in the next slides. So just looking at the same thing with reference to the diagrammatic representation that I have just tried to represent all the 6 types of reactions. So what is the oxidation reaction? Reduction reaction means there is either loss of the reactant loss of the electron or the loss of or the gain of the electron. So this is the simple reaction that which shows that there is a loss of electron from reactant A to gain of electron by the reactant B. So this is the electron acceptor, this is the electron donor ultimately. So these type of reaction they are called the redox reaction or oxidation reduction reaction. Second is the ligation. So if you look at the biochemical component, that there is a ligation means the joining. So joining of these two molecule pyruvate in the plus the carbon dioxide to form the oxaloacetate. So this carbon dioxide is getting joined over here in the presence of ATP, and this ATP is basically reduced to ADP and inorganic phosphate. So this ATP is basically used as a energy molecule for the formation of this product. Similarly, the isomerization. So isomerization is nothing but there is a functional group change. So let's say this is a OH group, this is a alcohol group. So this is getting transferred within the same molecule. So the function is getting the functional group is changed from one position to the another. And the example that comes, that's a very common example, is the formation of isocitrate from the citrate, right? So this is the isomerization reaction. Similarly, the group transfer. So this is a very common reaction that is going on or proceeding within the body. That formation, that the ATP is broken down during the formation of a very common cycle called the glycolysis, where glucose is converted to glucose. This is the very first step of glycolysis, that glucose is converted to glucose 6 phosphate in the presence of ATP. So normally we write this reaction as glucose to glucose 6 phosphate in the presence of ATP. So ATP is converted to ADP plus PI, and this phosphate, this is basically getting transferred to the glucose in the presence of enzyme hexokinase. So you can just clearly summarize, or you can just clearly think that this enzyme is basically helpful in transferring this phosphate from ATP to ADP. So it's a two-step reaction, even though we are writing it as one-step reaction. Similarly, the hydrolysis. So hydrolysis is nothing but the uh, water molecule. It basically splits the reactant into two products, right? Followed by the last type of reaction, or you can say this is one of the another components, another reaction that's called the glucose, this 2-phosphoglycerate. So formation of a double bond. So this phosphoglycerate to phosphoglycerate is converted to the phosphoenol pyruvate. So this is again a type of reaction that is going on within the body. Now the question that arises is why these reactions cannot be proceeded without the actions or without the presence of the enzymes, or why the enzymes are needed. So the question that I'm asking is that why the chemical reactions are not performed without enzymes or without the help of enzyme in the living system. So the answer to this question that lies is that most of the chemical reactions within the organism would be impossible under the condition because of the very first thing, that is the body temperature. Since the body temperature is not so high, and the reaction to proceed from A to B, it requires a lot of energy. So this energy in the chemical or in the chemistry on the lab, we can just use, you can heat the test tube and basically provide the extra energy that is required for the conversion of a substrate A to substrate B. So that is not possible in the living systems, and since the routine body, normal body temperature cannot go beyond a certain limit. So second thing is that reactant may be present in a low concentration, since we are aware that the various reactions, they are carried out in the presence of various metabolic pathways. They are carried out in the presence of certain cofactors, which are the components of our daily food, including your vitamins and other components. So it may not be possible that since they are present in a very low amount, so they are able to interact in such a efficient way that reaction can proceed in the spontaneous manner. So that is the primary concept or the primary logic that why these molecules or these reactions require the various types of energies, the various types of enzymes. So therefore, the rate of the most biochemical reaction need to be increased by the help of a catalyst, and this catalyst is nothing but it is a chemical that speeds up a chemical reaction. So in the living system, the enzymes, they are working as the enzyme catalyst, and they are called basically the biological catalyst. So this is the primary thing that before we try to look at the enzyme and its classification, we should be aware that what is the significance of using these enzymes in the biochemical reaction. Now coming to the definition of enzyme. So what is an enzyme? Enzyme is nothing but it is a protein in nature. That means it's a proteinaceous nature. That means it is made up of various amino acids. But besides the nature that it's a amino acid, can be a ribonucleic acid which is acting as an enzyme. So sometimes they are classified that these are, so they are called a special category of enzymes called ribozymes. So these ribozymes are nothing but they are derived from the RNA or the ribonucleic acid, and so those ribonucleic acids which are behaving as an enzyme, they are called ribozymes, and they are produced by the living cells. And the specific and the condition is that they are highly specific and they are highly catalytic to its substrates, and they are very much important in the macromolecular biological catalyst. So due to their specificity, they can and due to their specificity, and since they are able to carry out or they are able to proceed with the lowering of the energy, they are very efficient under mild environmental conditions. So coming to the history of enzyme and enzymology. Enzymology is nothing but it's a study of enzymes. So it's basically enzyme plus ology, which is the study of when you try to study the enzymes. So this is the called the study the enzymology, and enzyme is a Greek word which means in the living. En stands for in, enzyme is the living. So this is what is the definition of a enzyme. The Greek meaning of the enzyme, so it is basically acting as a biocatalyst or a organic catalyst which is having high molecular weight protein, right? And this term was coined by the term enzyme was basically coined by the scientist Kohne in the year 1878, and the first enzyme which was extracted was from the yeast cell by the Buckner in the year 1897. And first purified enzyme is the urease by the scientist James B. Summer in the year 1962. Even though the term was coined in 1878, and the first enzyme, exactly the enzyme was extracted by Buckner in the 1897, but it was reported that the study of enzymes was started way back in the year 1830, where this Davenport, they prepared a malt extract from the germinating barley seeds. So the history goes beyond the 1870s, where it was initially classified. So it is from the 1830 to 1833 and then 37. So various scientists during that time, they identified, or they basically looked after that there are certain things which are acting on the various biomolecules, even though at that time the classification was not so proper. So the posture also demonstrated that the alcoholic fermentation is brought by the action of the living cells, even though they are also not able to characterize that these are carried out by specific enzymes. And Summer, we have already discussed that they are the first one to extract the enzyme from the cells. Then coming to the classification point of view, that these enzymes are classified typically till the IUBMB, or International Union for Biochemistry and Molecular Biology, they propose a proper nomenclature when the number of enzymes start getting increasing. So earlier they were named just randomly on the basis of the type of their reaction they are been getting categorized. For example, the enzyme catalase or papain or trypsin or lysozyme. So they are just named because they are carrying out the specific type of reactions. Like catalase, they are involved in the dismutation of the hydrogen peroxide to water and oxygen. Similarly, pepsin, this is a endopeptidase which is breaking the, which is a protease. So enzymes are nowadays they are classified, or they are basically analyzed on the basis of their nomenclature. They are defined in terms of two ways. One is the tribal name, that is, which is a very common or everyday use name, which is called the tribal name, and they are always explained, or they are always named after this substrate or the type of enzyme they are getting, the type of reaction which are getting catalyzed, and they are ending with the term ages. So this is a common and recommended name versus the systematic name, that these enzymes are basically classified based on and basically they are named as this. Another classification that comes to the picture in the early 90s was the enzyme classification number. So what is this enzyme classification number? Enzyme classification number is a number that is given to each enzyme, and it has four different parts. We'll just study. And what is this EC number? So they are designated as the EC number or ECG classification. So before we move to EC number, just look at the common examples. That recommended name is for a particular enzyme. Let's say is the urease, which is the very first enzyme that is isolated. So it, as far as the reaction is concerned, it is catalyzing the hydrolysis of the urea, which is according to the type of catalytic reaction it is performing. And the systematic name for this urea is like urea amino hydrolase. So this is how the nomenclature was initiated. And nowadays there is another component, or what to say is that after the EC number was defined, so these are further characterized by the EC number or the enzyme classification number. So what is this enzyme classification number? So these enzyme classification numbers are basically subdivided into 4 digits. For example, enzyme classification with EC, that A dot B dot C and dot D, where A is called the class, B is a subclass, C is a subclass, D is basically a subclass, or basically the number of enzyme at the position, right? So I will explain this in the next slide, that what is this B and C. So B and C basically they are the digits that describe the reaction, where D is a digit is a number of enzyme of that same function, so based on the actual substrate. So let's take an example of this, like EC with 1, 2 and 3, 4. So class, so 1 means we are talking of the class. Let's say class in terms of 1, it's a oxidoreductase. So 2, if you look at 2 is a subclass. of the enzyme oxidoreductase, which acts upon the aldehyde. So it basically gives you information on the type of reactant they are getting involved. In subclass 3 is that this is a subclass that this aldehyde or this reaction is preceded by in which the oxygen is acting as an acceptor molecule, and D or the fourth number is a serial number of within this subclass. So this is how we classify the enzymes. So as far as the IUB or IUBMB classification, there are 7 different classes. Initially it was reported that there are 6 classes that were reported, but in the year 2018 a new class of the enzyme was added to the list of the already existing 6 classes, which are basically class 1, which is the oxidoreductase, B is a transferase, C is a hydrolase, D is lyase, isomerase, ligase. So these are the 6 classes that are already there, or they have basically characterized in the initial. And the 7th class, this class was added in the year 2018. So this is either initially it is called IUB, International Union of Biochemistry, and later it was named as IUBMB, International Union of Biochemistry and Molecular Biology. So first digit, which is a main class type, which include the type of reaction, that is we are talking of oxidoreductase. Second digit, subclass, type of group that is involved. Third is the subclass, which is a type of substrate on which the enzyme acts. And fourth is the fourth digit basically includes the enzyme, the systematic serial number, and the name. So these are the 7 different classes based on the which was defined by the enzyme IUBMB. So this is the latest information that EC7, which is a translocase, has been added as far as the information. So much more information you can get it from this link. So as far as the classification, I have tried to characterize, I have tried to basically depict the different classes on the basis of what is the class 1, which is the oxidoreductase, 1.1, which is acting upon CHOH group of donors with 1.11 bond, means it's talking of NAD or NADP as acceptor. So here again 1.1.1.1, so this is the exact name that is, it's a alcohol dehydrogenase. So we can write alcohol dehydrogenase as a tribal name, or EC number 1.1.1.1, or similarly 1.1.1.2 as it's a alcohol dehydrogenase. But 2 is that it is involving not NAD but NADP positive. So this is how the various classes are identified, or they are characterized. So this table is a extensive table you can study. So what I try to list is the summary of the various enzymes. That oxidoreductase, this is the type of reaction. A is reduced to A reactant A and B, they are involved in the reaction, and this is the ultimately A is getting oxidized and B is getting reduced by the transfer of electrons from A to B, right? So this is what is going on in the oxidoreductase. The examples, the most common examples are the dehydrogenases, oxidoreductase, peroxidases, monooxygenases, or deoxygenases. Similarly, we have transferases, in which the reactant A plus B, when it reacts with the C to form that, this reaction will transfer from A to B, A to C, sorry. So this is the examples, where it's aminotransferases or phosphotransferases or glycosyltransferases transferases. That means, as the name indicates, just looking at the name, you can just clearly indicate that this glycosyltransferase means there is an involvement of the glucose moiety which is getting transferred, or the amino moiety is getting transferred from one reactant to the another. Similarly, hydrolysis. So in the presence of this water, the reactant, one of the reactant is getting hydrolyzed. So the examples are this peptidases, amidases, glycosidases, esterases. So the list is endless in all the categories. So depending upon the area of research, area of study, or the extent we want to study, we can remember or we can use this classification. Similarly, the class lyase or synthesis, which is A plus B. So that means there is a joining of the two. So reactant A and B are getting joined. So this is the example of the subclasses: carbon-carbon lysis, carbon-oxygen, carbon-nitrogen, carbon-sulfur lysis. Similarly, the isomerization or the structural conformation change from one reactant to the another. So this is what is the examples of isomerases, cis-trans isomerases. So there are so many examples of very important components in the biological biochemistry that various metabolic reactions are basically carried out using all these types of reactions. Similarly, the ligases. So ligases are also called the synthetases, and they are different from the synthetases that they require ATP as one of the energy molecules for joining. Even though these and this both lyase and ligases they are almost performing a similar function, only difference is that these ligases they are involving the ATP molecule, the energy molecule as such, for the transfer of or for ligation of the two molecules or joining of the two molecules. So this is what is the example like carbon-carbon ligases, carbon-oxygen, so on and so forth. So similarly, the new class which is added, they are called the transphorases. So in this case, what is the example? The component is that there are two reactants. So this is getting transferred to this A, and this just we have to look at that this is on one side. So after the transfer, this is going to be in the second side. So I will give you the example in the next slides to give you the information on what is the mechanism, and this is the example like ABC type phosphate transporters, or ABC type sulfate transporters, or glutathione transporters. So these are various types of transporters. Now coming to the various examples. So oxidoreductases and transferases. So what we have seen that ethanol plus coenzyme, which is NAD, like we have already seen that this NAD belongs to category 1.1.1.1. So it's alcohol dehydrogenase. So this ethanol is converted to aldehyde, and this is getting NADP positive, DH positive. So these two H ions, one from here and another from here, they are getting dissociated to form this. Similarly, transferases means there is a group transfer. So if you look at the example of alanine plus alpha ketoglutarate to form the pyruvate and glutamine, so what is happening is as far as structure is concerned, this amine group is getting transferred from alanine to alpha ketoglutarate to form glutamate, and this is converted to the pyruvate. Similarly, when you talk of hydrolysis, so hydrolysis means they are breakdown. So this is a example is the peptidase here. So this polypeptide with C terminal. So this C terminal is broken down into the shorter peptide and the amino terminal from the amino acid from the N terminal. They are getting separated. So this is the C terminal component. The C terminal amino acid. So this C terminal amino acid is getting dissociated in the presence of the enzyme peptidase. So typical subclass peptidase lipases or amylases. So even we talk of lipases, that means we are talking of the lipid breakdown or the amylase. So it's a amylose group that's getting broken down. And what is the type of function they are performing? This is there. Similarly, when we talk of lyase, then there is a addition of a group to a double bond, or a removal of a group to a double bond. So like for example, pyruvate is converted to aldehyde and carbon dioxide. So this H is getting attached, ultimately leading to, so this H is basically attaching to here, and the carbon dioxide is getting separated. Similarly, isomerase and the ligases, these are the examples that isomerases, you know the structure of a malate. So this mallet has the two carboxyl, the CO negative groups on one side. So after the isomerization, what will happen that this CO group, it will be shifting from this position to this position. So that becomes the fumarate. And similarly ligases, so in the presence of ATP, this pyruvate is converted to oxaloacetate in the with the release of ATP. So this ATP is basically used as the energy molecule that proceeds this reaction. Right? And the last category in this case is the translocase, which is a new category which is defined. So in this case, there is an example I will give you is the ATP ADP translocase, and this ATP and in the presence of water, the H ions they get translocated from one side. Basically, it's a transfer across the membrane. So ATP it is transferred from one side to the another, or so this is a ADP ATP translocase, or there is another component which is a phosphotranslocase which are transporting the phosphate molecule across the membrane. And the various examples with this is basically with reference to your electron transport chain. So what you can say that as far as the latest information is concerned, these are the 7 classes that are identified by the IUBMB for the classification of enzymes. So I hope you people have understood the concept. So thank you very much.